Good afternoon. I'm Luca Alberici. I'm the head of the Cell and Gene Therapy Division at AGC Biologics, and it's a pleasure for me to have the opportunity to present you AGC Bio, your friendly CDMO expert. So let's start. First things first, our mission is as many to bring hope to life, but this is not just paper, just words on paper. This is obviously a value that we really live through on a day-to-day operation, from the leadership team all the way to the technicians. To save life for the last 30 years has been really on top of our mind, and we are happy that we have been very successfully helping this industry to move forward. As you were reading in the first slide, we are calling ourselves the friendly CDMO expert. And why? Because we are living, unfortunately, in the industry where there are a lot of CDMOs, but they are not really always constantly delivering. Some of them they know what they're doing, they've been around for a long time, but they're very hard to work with, especially the big ones. Others tend to be a bit smaller and nicer, but not to know exactly what they're doing because they're too new. What makes us a bit unique is the fact that we have been around for 30 years, and we have delivered many products to the market, but at the same time we maintain a size and a mindset that help us to be easy to work with and to at the same time deliver the manufacturing to our customers. Everything starts with being part of a large Japanese conglomerate, as AGC Inc, that most of you might not know, but it's probably the leading providers in glass. And by operating in a very large and commoditized industry, it provides cash, it provides long-term vision. That is what allow us to be around for long-term and being very much long-term oriented, so don't need to look at the economics of the year, but building something with our customer and partners throughout for a long-term journey together. We operate across many modalities, protein, both on mammalian and microbial platform, nucleic acid on mRNA and DNA, and of course, viral vector and cell therapy. That is the division that we are going to cover today. On the right-hand side, you see some numbers that tend to speak by themselves. So we have obtained 10 commercial approvals to date. That is probably the largest number of commercial approval any CDMO has obtained, with 6 viral vectors and 4 cell therapy, and last but not least 2 DNA, and of course hundreds of batches manufactured in viral vector and cell therapy, and currently serving over 30 clinical trials. We operate a network of 8 different facilities, pretty equally scattered around the globe, 2 in US, 2 in Asia, and 3 in Europe, with centralized manufacturing for viral vector in Milan, and 3 sites for cell therapy, each of them in a geography, ensuring a good local coverage, especially for autologous therapy. So 1 in US, 1 in Europe, and 1 in Yokohama. But let me go in the details of those. So Milan is the biggest site in the network, with around 360 people working there. Here we manufacture vectors and cells. This is the only facility in the world who has received a commercial approval by both EMA and FDA for both viral vector and cell therapy. And on top of that, we also had the latest program was the ICMC certification by Dark Horse. This is also the oldest facility with 30 years of experience, and 10 commercial approval that I was mentioning. They are all supplied from the facility in Milan. Viral vector-wise, we cover retro, lenti, and AAV, with scales that range from 50 liters all the way up into 2,000, with the sweet spot being somewhere between the 50 and 500 liters. We do both adherent and suspension based, and I will come back to that later on. And for cell therapy, we also have quite a lot of experience on T cells, hematopoietic stem cells, NK, MSCs, and lately getting in the iPS world. Another key feature of this facility is the extensive QC expertise, and that allows us to test over 160 different testing houses, so relying very little on external testing labs. Moving to east, we go to Yokohama. That is the newest facility in the network that is currently operating 2 Grade B suites and for clinical trial, and is currently under construction, the commercial facility in Yokohama with 4 additional Grade B suites for both autologous and allogeneic cell therapy. Here in the US, we have had for the last 5 years a facility in Longmont, Colorado, that has been our center in the US for cell therapy as well. We currently have 3 Grade B suites of around, let's say, 50 to 60 square meters each, that all of them can manufacture both allogeneic and autologous products. And last but not least, in Germany, in Heidelberg, we have our Center of Excellence for nucleic acid, especially DNA, pDNA, and mRNA, with scales that range from 10, 50, and 100 liters at both R&D, high quality, and full GMP grades. And here, I was alluding to before of our 10 approvals. Here is actually you can find by our green logos next to it, all the products that we supply that have been currently approved by either Europe, US. Some of those we manufacture viral vectors, some other the cell therapy, and in most of the cases we actually manufacture both vector and cells, making us also by the number of approved therapies the largest CDMO in cell and gene to date. And our late-stage focus is even further reinforced by our experience in validation exercises and PPQs. Here is an example of the fact that we already completed 13 PPQs, and we have 5 coming up between the end of this year and next year. And so we have basically validated processes for 10 years now, of which many have already reached the market and a few others are coming up in the next couple of years, making us again among the very few who not only have brought product to the market, but have done consistently multiple times. So. Now let's focus the remaining part of the presentation on the 2 pieces, vector and cell therapy. So vector-wise, we do offer proprietary platforms. You see this Pronto LVV and Bravo AV are our Lenti and AAV platforms. What do we mean by platform? It means that when you walk in with your gene of interest, you can find at our premises already a process running, the full set of analytics, reagents, meaning master cell bank, plasmids, and regulatory supports that allow you basically to just not having to deal with a lot of manufacturing issue and just rely on the expertise that was created in our premises in the last several years. As I mentioned, we have different scales. We can start low, as low as 50 liters, both in adherence through cell factory that can be then scaled up to fixed bed bioreactor like the SLS 500 by Cytiva, or suspension, serum-free classical suspension process ranging from 50 liters all the way up to 2,000 liters that has been already scaled up all the way into 1,000 liters bioreactors. Let me go back to this. So when the expectation is that you can, from the date of engagement, get the first GMP batch fully released within the first year. thanks to leveraging pre-existing data and platform. And we also offer, as you see here on the slide in the black box, CMC Jumpstart. That is basically a package of feasibility work that allows any developer to test us before fully committing into the, let's say, larger scope and potentially GMP manufacturing. So test us first, see whether what we claim is actually true, and then engage us for the full deal. And this program has been extremely successful, allowing to lower the entry commitment and to test at first. So I was mentioning that we not only have, obviously, the full-scale range of services and equipment, but we also have a full range of scaled-down models, fully representative, that allows really to get representative data and prediction of how the final scale is going to look like. And this is important because one of the key selling points that we are promising the industry is that we are able to lower the cost of goods, especially for lenti, where there are claims around that can cost 30,000 euros per patient, 50, 70s. Well, what we have delivered to date pretty consistently, and these are actual numbers from current customer, we are able to deliver lenti for a price per patient that ranges between 4,000 and 8,000 today, using our smallest scale, so around 50 liters. That price can go down into the range of 2,000 to 4,000 when using around 200-liter scales, but it can be further scaled up to 1,000 liters, allowing to reduce the cost on a per-patient base of our viral vector all the way into 1,000 euros or less. And looking to cell therapy, we have an installed park of equipment that is pretty broad. Most of them is currently on site. Others have been tested or have been honed previously and are really process specific. But we do have use and experience on most of the commonly used equipment in the cell therapy field. And we also work on different models, from slot models, dedicated suite, and rental model option. Let me give you a bit of a more sense of what do I mean. So slot models, or generally dedicated rooms, what does it mean? It means that we have like small grade B suite. This is mostly used for open processes where you need hood, centrifuge, and not a lot of closed steps, and therefore the suite is entirely dedicated to the patient throughout the entire duration of the process. We can have slots, or we can have a customer basically renting the entire suite for a period of time, allowing more flexibility in the planning. We have 6 of them, for example, in Milan, and 3 of those in Longmont, and another 2 in Yokohama. So overall, we have around 10 suites that are allowing to do open processes. This is another example. This is again out of our Milan facility. This is a ballroom where we can install up to 12 prodigies to date, so allowing up to 12 CAR-Ts or TCR-Ts manufacturing in parallel. And as you can imagine, this allows a throughput in the few hundred of patients per year base. And last but not least is a dense floor model. That means that each of the different unit operation is segregated in a different rooms that allows a better focus and an optimization of that specific unit operation. Here is again an example of an allogeneic process that we were running in our facility, where the 3 different steps of the isolation, the expansion and genetic modification, and then fill and finish were all segregated into 3 different suites in order to optimize the throughput. And with that, thanks for your attention. There are a few of our BD's people around, so don't hesitate to reach out to me, to visit the AGC Bio site, or to reach us through the partnering system if you want to know more about our commercial experience, about how we would handle potentially your program. Thanks a lot for your attention.
Thank you, AGC Bio.